Let us move on to the next non-Mendelian inheritance, which is called as the codominance. So till now we were discussing about the crosses where the F1 generation resembled either of the two parent, which we saw in dominance, monohybrid dominance. Mendel cheyda experiment he got one of the two parents trait, and or the other one which we studied was in between. That was the trait of both the parents were not seen, they were in between the both parents characteristic. Okay, red and white, it gave rise to pink, so it is called as incomplete dominance. But here in this codominance, these are the cases where the F1 generation will resemble both parents. രണ്ട് പാരൻസിനും എന്ത് ട്രേറ്റ് ആണോ ഉള്ളത് അത് രണ്ടും എക്സ്പ്രസ് ആവാൻ ചാൻസ് ഉള്ള ഒരു ഡോമിനൻസ് ആണ് കോഡോമിനൻസ്, right? So it is the inheritance in which both alleles of a gene are expressed in a hybrid. Okay, in an offspring we can see the expression of both the alleles of a single gene. ഒരു single gene രണ്ട് alleles express ആവുന്നത് we can find it here. Right, and a good example is different types of red blood cells that determine ABO blood grouping in human beings. So you'd have heard about blood groups, right? Different types of blood groups which human beings have. So we will be learning about the example of how different types of red blood cells which determine the ABO blood grouping in human beings. Right, so in our human body ABO blood groups are controlled by the gene which is called as I. I എന്ന് പറയുന്ന ജീനാണ് നമ്മളെ ബ്ലഡിലുള്ള ബ്ലഡ് ഗ്രൂപ്പ്സിനെ കൺട്രോൾ ചെയ്യുന്നത്. ഓക്കെ, സോ യൂസിങ് I only we will be denoting all these blood groups. Okay, so the plasma membrane of the red blood cell has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. അപ്പോൾ എങ്ങനെയാണ് ഈ I എന്ന് പറയുന്ന ജീൻ കൺട്രോൾ ചെയ്യുന്നത്? It is done with the help of the antigens which are present. So these are all antigens. You can see the red blood cell, biconcave red blood cells. On their plasma membrane you can find sugar polymers which is protruding from its surface. So the outermost surface is the plasma membrane, and from there you can see many sugar molecules which is protruding, right? And the kind of sugar is controlled by the gene. So ഇവർ എന്ത് type of sugar ആണ് produce ചെയ്യുന്നത്, that is depending upon the gene which is controlling it, which is I gene. So I gene ആണ് എന്ത് type of sugar ആണ് അവർ produce ചെയ്യുന്നത് decide ചെയ്യുന്നത്. Okay, so it controls the production of sugar polymers, that is antigens that protrude from the plasma membrane of the RBC. Clear? Now use a common term called as antigen for this. No need to confuse it with sugar polymers and all. Just keep as antigen. Think about an antigen is present on the plasma membrane of the red blood cells, right? And this the plasma membrane which contains this antigen is controlled. Their sugar production is completely controlled by the gene called as I, right? The gene which is I, capital I, has 3 alleles. How many alleles are there? So usually we have only 2 alleles for a particular character, but this gene called as I, it has 3 alleles. That is I capital A, I capital B, and small I. Okay, so this gene has 3 alleles which is capital I and capital A, capital I and capital B, and small I. Right, and in this the alleles called as capital A and capital B will produce a slightly different form of the sugar. Okay, so they are same genes, but the sugar which they produce is slightly different. That is why they are named as A and B. Right, so these two will produce a slightly different form of the sugar, and allele called as small I, it does not produce any sugar. Okay, so that is the difference between IA, IB and small I. Clear? And we know that human beings are diploid organism. Appo all these 3 alleles will not work in human being, only 2 of them will be seen. And these 2 alleles which the diploid organism have, it depends upon what they get from the parents. എന്താണോ പാരൻസിൻ്റെ അടുത്ത് നിന്ന് കിട്ടുന്നത് അതുപോലെ ആയിരിക്കും അവരുടെ അലീൽസ് ഉണ്ടാവുക, റൈറ്റ്? സോ 3 different alleles are there, but as organism called as human being is diploid, it will have only 2 types of alleles in one organism. right? So because humans are diploid organism, each person will possess any two of the three I gene alleles. Okay, so either you will get capital A, that is I capital A, or you will get I capital B from one parent, and again same way from one parent you will get I capital A or I capital B or small I, same way with the other parent also. ഇപ്പോൾ രണ്ട് പാരൻസിൻ്റെ അടുത്തുനിന്നും ഒന്ന് capital A ആയിരിക്കാം, അല്ലെങ്കിൽ capital B ആയിരിക്കാം, അല്ലെങ്കിൽ or it can be O. Okay, so O is nothing but it does not produce any sugar, right? So it is like null, zero എന്നുള്ളൊരു അർത്ഥമാണ് O എന്ന് പറയുന്ന ഒരു ഗ്രൂപ്പായിട്ട് നമ്മൾ പ്ലേസ് ചെയ്യുന്നത്. Okay, so from one parent you will receive only one of the allele, and same frame from the other parent you will receive only one of the allele among them. And which are the three alleles? I capital A, I capital B, and I O. Or it can be written as small I also. Okay, so in your textbooks it is written as small I, you just learn it like that. Okay, now let us see the expression of the blood groups based on the alleles which they receive. So always keep in mind, I capital A and I capital B are completely dominant over this small I. Okay, because I capital A and I capital B will produce sugars, but small I, it does not produce any sugar. അപ്പോൾ എപ്പോഴും ഏതായിരിക്കും dominant? I capital A and I capital B will be dominant, okay, over the allele which is called as small I. Okay, so this can be explained. I will just explain it with this, but keep in mind that only I capital A and I capital B are dominant and small I is always recessive. It is because they do not produce any sugar. Okay, now let us see how these alleles are working. Okay, so let us see how this IA and IB is dominant over I. Let us see how we receive the alleles from the parent and what is getting expressed inside the human body. So first we will see the alleles from parent 1. We can take parent 1 as 1 and 2. Male can be taken as 1 and female can be taken as 2, or else in either way. Okay, so we will find out the genotype of offspring and also the phenotype. Okay, and phenotype here means the blood group which the offsprings possess. So suppose if, keep in mind, capital A and capital B is dominant over the small I. That is, I capital A and I capital B will be dominant over the small I, which means that in the presence of small I also, only the dominant gene will express. See this, capital A, so you are receiving one, from first parent, that is male parent, if you are receiving capital IA, and if you are receiving and same allele from parent 2, which is the female, that is capital IA, it gives rise to the genotype which is capital IA, capital IA. Okay, so the alleles formed will be capital IA and that capital IA, right? And the blood type which the person will have is A, because these are dominant alleles, so it will form A. They both code for the same character also, so it is A, right? And if you take the case where you receive capital A from one parent and small I from another parent, so think that from your mother you have received small I. We know that capital A is dominant over small I. Okay, so their genotype will be capital A small I and their phenotype will be capital A, because I is recessive, the small I is recessive, appo adu express avula, only the capital A gets expressed. So the offspring will have capital A as their blood group. Okay, now coming to the same thing, if capital B is from one parent and again from the second parent also, if it is capital B which we receive, then the genotype will be capital B, and the phenotype will be B, which means that the blood group will be B. Okay, and if you take that from one parent, if it is capital B, and from another parent if it is small I, then it gives rise to a genotype that is capital B small I. And we know that capital B and capital A are always dominant over small I. So this small I will not get expressed and it gives rise to blood group B. Okay, now coming to the next type where this codominance occur. അപ്പോൾ നമ്മൾ ഈ codominance example ആണ് ഈ blood group പഠിക്കുന്നത്. In that we are going to learn about the codominance now. Suppose if your father has capital A and if your mother has capital B, the genotype which it forms is capital A and capital B. And the phenotypic appearance, which means the blood group will be, what is it? It will be AB because A and B both are dominant. Okay, so both is getting expressed in one single person. But രണ്ട് alleelsum എന്തായിട്ടുണ്ട്, രണ്ട് different alleelsum they have got expressed. So it is called as codominance. So they are working together. Okay, so that is why it is called as codominance. And again reverse one, if from male parent if you are receiving B and female parent if it is A, again it combines to form IA, IB and it gives rise to blood group which is AB. See, ഞാൻ പറയുന്ന capital A or IA both it codes for the A blood group. Okay, I'm just saying A in somewhere and IA in some places, right? So same thing is same, but I'm seeing it in two ways. Okay, so the blood group which it gives rise is to AB again. You can see the blood group here. So this is the blood group A. You can see the antigen, its structure is different it is different from the structure of blood group B. And same way you can see here AB blood group, it has both the antigen, but you can see O, it does not have any of this antigen. Okay, so small I, if a parent is giving small I and if another parent is also giving small I, it gives rise to recessive trait which is the O. Okay, so when capital A and capital B are present together, they express their own types of sugar and this is termed as the codominance. Clear with the children? Okay, so since there are 3 different alleles, there are 6 different combinations of these 3 alleles. Okay, 6 different combinations are namakku kittuvana and that are possible. A total of 6 different genotypes of the human beings is found. Right? So 6 different types of genotypes are seen and same way 6 different types of combinations can be given. So 6 different combinations and 6 different genotypes. How many phenotypes are possible? Only 4 phenotypes are possible. It can be A blood group or B blood group or AB blood group or O blood group. Right? So clear with this codominance, children? Okay. If you see the example of this ABO blood grouping, it is giving a good example of multiple alleles. Right? So we are able to find how the multiple alleles will work. Right? So as we all know, there are more than 2, that is 3 alleles in this multiple alleles. രണ്ടിനേക്കാളും കൂടുതലാണ് ഉള്ളത്. Okay, which governs the same character. And since in an individual only 2 alleles can be present, multiple alleles can be studied only in a bigger population. Okay, ഒരു person എടുത്താൽ ആ പേഴ്സണിൽ ഏതെങ്കിലും 2 type of alleles മാത്രമേ ഉണ്ടാവുള്ളൂ. So this multiple allele concept can be found only when the population studies are made. Okay, now let us move on to the last part of this codominance. Now let us see about the last example in the case of incomplete dominance. So occasionally a single gene product may produce more than one effect. Sometimes a single gene product may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. Okay, അവരെ കൺട്രോൾ ചെയ്യുന്നത് ഈ സ്റ്റാർച്ച് സിന്തസിസിനെ പീ പ്ലാന്റ്സിൽ കൺട്രോൾ ചെയ്യുന്നത് ഇറ്റ് ഇസ് വൺ ജീൻ ആൻഡ് ഇറ്റ് ഹാസ് അലീൽസ് 2 alleles, one is capital B and the other one is small b. And starch synthesized efficiently, the starch is synthesized efficiently by capital B gene, and therefore large starch grains are produced by this homozygous capital B. And if you take in contrast the small b which is homozygous have lesser efficiency in starch synthesis. Okay, it means that they will not be very efficient as like the capital B, and they produce the smaller starch grains. Okay, so they produce smaller starch grains. Clear? Now, after maturation of the seeds, the capital B are round and the small b seeds are wrinkled. because here the efficiency of producing starch is lesser than capital B, capital B. And if you take the case of this heterozygous condition where there is capital B and small b, in that condition the heterozygous will produce round seeds, and so B seems to be, that is a capital B seems to be the dominant allele. Okay, so as the heterozygous seed is also, heterozygous seed, it is producing the round seeds, which means that capital B seems to be the dominant allele. But the starch grains produced are intermediate size in the capital B, small b. Right? So their seed will be round, but their size is intermediate. It is not as like the starch which is produced by capital B, capital B. So you can see here effective starch synthesis is by capital B, capital B. It produced large sized seeds, and lesser starch synthesis is seen in small b, and small sized seeds are produced. And when you take this heterozygous condition, the seeds will be round here. Okay, it is not wrinkled. So in this capital B, the seeds produced will be round, and here it will be wrinkled. Okay, so in this heterozygous condition, the seeds will be round, but it does not have as much starch grains produced by capital B, capital B. So they are intermediate sized. Okay, nalla velipam undavilla, bayangara wrinkled aayirikkilla. They have intermediate sized. So if the starch grain size is considered as the phenotype, then from this angle the alleles will show incomplete dominance, because along with the dominant trait the recessive one is also getting, that is new character is also getting expressed, not the recessive one, the new character is also getting expressed, which is the intermediate sized seeds. That is called as incomplete dominance. Okay, so from this we can understand dominance is not an autonomous feature of a gene or the product that it has information for. Right? So it is not dominance, it is not an autonomous feature. What is this autonomous feature? It is nothing but the to control something. Right? So dominance is not an autonomous feature of a gene or the product that it has information for. So it thus depends on much on the gene product and production of a particular phenotype from this product, as it does on the particular phenotype that we chose to examine, in case more than one phenotype is influenced by the same gene. അപ്പോൾ നമുക്ക് പറയാം ഈ dominance എന്ന് പറയുന്നത് ഒരു what to say, it is not an autonomous feature, which means that it does not have a governing rights there, right, of a gene or the product that it has information for. So dominance will never be an autonomous feature of a gene or the product which they produce. All this will depend on, the dominance will depend as much on the gene product. ആ ജീൻ എന്താണോ പ്രൊഡ്യൂസ് ചെയ്യുന്നത് അത് ബേസ് ചെയ്തിട്ടാണ് ഡോമിനൻസ് ഉണ്ടാവുക, ആൻഡ് ദ പ്രൊഡക്ഷൻ ഓഫ് എ പർട്ടിക്കുലർ ഫിനോടൈപ്പ് from this product as it does on the particular phenotype that we chose to examine. അപ്പോൾ നമ്മൾ എടുക്കുന്ന ഫിനോടൈപ്പ് എങ്ങനെയാണോ അപ്പിയർ ആവുന്നത്, it is based on that their effect is seen. Okay, in case more than one phenotype is influenced by the same gene. So from this incomplete dominance we can find that more than one phenotype is influenced by the same gene, because we saw here ഒരു particular gene തന്നെ capital B തന്നെ it is appearing large sized in homozygous condition, and in heterozygous condition it is not as much large, it is intermediate sized seeds. Okay, so this is about the non-Mendelian inheritance, children. So I hope you are clear with codominance, codominance and the that is incomplete dominance, and I hope you are clear with dominance also, which was the Mendelian's experiment. So please read your textbook properly. If you have any doubts you can ask to me. So please go through it. You have MCQ questions this time. For your first time you will have MCQ questions, so line by line it is very important to learn and understand it. If you have doubts please ask to me. Okay, so Om Namah Shivaya. Thank you.